Compass has been giving us a lot. And one of the things Compass helped us out with was mapping the somatic mutations in newly diagnosed patients. And that helped us design a new umbrella trial. It's called MyDRUG umbrella trial. It's a multi-arm study, and that's on the basis of activating mutational patterns that we observed in newly diagnosed patients. And one such arm of MyDRUG umbrella trial that I focused on my talk at IMS was on an arm that identifies patients with activating RAS and RAF mutations and treats them with MEK inhibitors such as Cobimetinib. So in this case, again, born out of discoveries from Compass, we identified patients with activating mutations, treated them with MEK inhibitors, and obtained their tumor microenvironment from the bone marrow aspirates, and studied it using single cell technologies. And we were able to map out some of the cell types that will give us more insights on how MEK inhibitors are functioning. MEK inhibitors interact with tumor microenvironment, not just the tumor itself. And we were able to see some patterns that will perhaps help us understand the underlying biology driving the response to MEK inhibitors.
Can you say a little bit about the scale of that study?
Yes, so there were 9 patients that were enrolled in MyDRUG that had at least one sample obtained at baseline. And the patients were treated 2 cycles of Cobimetinib and Dex, and following which they received backbone therapy. So at least one patient has sample obtained at baseline, so which means we have like 9 patients with single cell data. So that enabled us to look at least about more than 50,000 single cells. But to put things into context, for the purposes of the study, we made use of existing external data sets that describe normal bone marrow and newly diagnosed multiple myeloma. So we integrated these data sets to give a context to our discoveries. Because then we'll be able to say, if we were able to identify a pattern, whether the pattern has been observed in normal bone marrow, followed by newly diagnosed multiple myeloma patients, and how is that changing or being affected by MEK inhibitors. So in the study, we made use of a few external data sets, combined them with our data set, and then we were able to tease out particular T cells and monocytes that were previously described in the Immuno-Atlas study. And we were able to see some patterns, very small patterns associated with MEK inhibition. So, George, where do you think the technology is going to go from here?
Well, I think what's exciting about this capability to understand the immune system now is we're in the era of immune therapy for multiple myeloma, and we can now begin to decipher the clinical effects in the context of whether a patient's immune system was fully healthy or partially unhealthy or fully inactivated. And so we're excited about these breakthrough studies that we've discussed and shared today, but it really goes forward throughout the MMRF programs, and I'll mention just a couple ways. One is that we consistently do retrospective studies from partner groups that may have sample sets that are particularly interesting clinically, and we can now apply this technology to those studies. The second is we'll do prospective translational research in order to understand specific immune therapies that are used in relapsed myeloma and to apply what we know now about the tumor biology of the patients and also their immune system, and integrate that to understand these clinical outcomes that we see, which are so promising, and yet we don't have really a handle on a cure at this stage. So we're driven to pull those two things together and optimize the therapy that we're now developing. And then the third case is that the MMRF Foundation is fortunate to have a clinical consortium in which we execute prospective interventional trials with a core group of clinical sites. And so this type of science will be fully integrated into those clinical studies. And we're preferring going forward now an adaptive platform that cycles in several different potential new interventions, and we can then read out not only whether they were highly effective or perhaps less than was hoped, but also see if that clinical result applied really only to certain subsets of the patients. So that's the most exciting part, I think, about how we're at the really early stages of immune science and applying it to clinical outcomes.